Welcome back to Molecular Biology and Biotechnology with Lucy. This is part 2 of lesson 1, which we are talking about the basics of molecular biology. And we stopped at cellular molecules. So cellular molecules, which are also called biological molecules, or in short, biomolecules. So these cellular molecules are small organic substances in the cells, and we have 4 main families of these biological molecules. We call them polymers because they are units, many units that are formed from monomers or building blocks. So we have polysaccharides, which commonly are called carbohydrates. Then we have fats and lipids. Then we have proteins, and we have nucleic acids, which we say these are mainly the DNA and RNA. So we have said they are polymers because they are made up of other smaller building blocks called monomers. So polysaccharides are made up from monosaccharides, or called sugars. For example, glucose. Glucose is a sugar or monosaccharide that makes up polysaccharides such as starch, etc. We have fats and lipids made from fatty acids, and glycerol. No fatty acids are important for making membranes of the cells. We have proteins, they are made up from amino acids. And we have nucleic acids, which are made up from nucleotides. So these are the 4 main families that form our biological molecules. Now from here, we are going to define what is molecular biology. Molecular biology, which is an intersect or a field that overlaps into 3 other studies. The first study is called cell biology. The other field is called biochemistry. And the other field is called genetics. Cell biology is the study of processes and structures that will support cell function. Biochemistry will study the chemistry of living systems. For example, what are those chemical bonds that are involved? What are the chemical groups? Is it carbon? Is it hydrogen? And the genetics is the study of heredity or inheritance in short. So these 3 are very key in molecular biology because it is the study of all of them in one that will help us to define what molecular biology is. So as a summary, we can say that molecular biology is the study of, remember we said it's the study of molecules because of this word. So mainly the molecules that we study in molecular biology are the DNA, so the study of DNA, RNA, proteins, and other macromolecules that are involved in cell function and heredity. And heredity is huge. We look at genetic variations, biodiversity, and so many others. So that is the study, and this study will use advanced scientific tools to be able to, extract some of these molecules. We'll look at how do you extract DNA, RNA, and proteins from the cells. How can you manipulate them? How can you image them? and finally analyze them so that we are able to apply them in health, in agriculture, in animal husbandry, and in many other functions. So that is molecular biology. And then we had mentioned that biotechnology actually comes from molecular biology, so that these concepts are now going to be added engineering so that we are able to form products that are useful. So biotechnology is now the combination of these concepts that we get from molecular biology in combination with engineering concepts, so that we're able to come up with products. We will go defining them and even understanding them further as we get more examples. Then now we go to something we call the central dogma of molecular biology. Central dogma of molecular biology. And this can be defined as a process. It's a process where we have the instructions in DNA being converted into a functional product. So the instructions in DNA being converted into a functional product. So we start from DNA. So our DNA normally, we will be looking at it further, but it's normally a double helix. That's single. So it looks something like that. My diagram is not the best. It looks like that. That is our DNA. Let's do it this way. That's DNA. So DNA comprises of information or instructions. For example, let's say that region wants to code for a certain product that is useful in the body. So for it to be able to convert this information into a product, this process will go into RNA, or the instruction will go into RNA. RNA is a single strand. That is our RNA. And then finally, it will go into a product, a protein. So now what will happen is this region will code for an RNA molecule, then it will finally code into a protein. Most of the time, our functional product is a protein, but sometimes it doesn't have to be a protein, because there are other regions. There are other regions in our DNA. For example, maybe we have a region here. That region is a non-protein coding region. It is likely to code for another product called functional RNA, which is a functional product, which is the final. So sometimes it doesn't have to end up here, but we will look at the reasons why. But generally, we are saying that this central dogma of molecular biology refers to the process in which the instructions from our DNA are converted into a functional product, which goes to RNA and finally goes to protein. And this whole process is also called gene expression. Gene expression. Very important. That process of converting instructions of DNA into a functional product is called gene expression. Now, this between here and here, this process of converting RNA to DNA has a name. It is called transcription. Transcription. It is called transcription. And then this process here of converting RNA into protein is called translation. Translation. So the process of converting DNA to RNA, transcription, RNA to protein, translation. And then we also have DNA multiplying itself into many copies of it the same. which usually is called replication. Now, this central dogma was defined by someone called Francis Crick in 1958, and he's one of the people who are able to define the structure of DNA as we will be looking at it. So that is the central dogma of molecular biology. We now move on to what are the applications, some of the applications of this molecular biology, or why should we study molecular biology. Applications of molecular biology we have in human health, in forensics, in agriculture, in animal husbandry, in ecology and environment, and we have others. In human health, it is used in detection of diseases such as cancer, genetic disorders, bacterial and viral infections. It is also used in drug design, that is in how to design medications that will target specific areas of our bodies to be able to cure the disease. It is also used in understanding the issues of heredity and inheritance, why you look like your parents or why you don't look like them. And it is also used in understanding several mechanisms such as antibiotic resistance, when we overuse antibiotics, behavior, and why some people are affected by diseases in a certain way. In forensics, it is used in identification of persons or individuals through your fingerprints. It is also used in paternity cases where we have a child being who is the father of a certain child. It is also used in crime investigations by trying to pick whatever molecular substances are left in a crime scene, like hair and body fluids. It is used in agriculture to detect pathogens that affect plants. It is also used in plant breeding. It is also used in making of genetically modified plants that can be able to resist, for example, arid conditions. It is also used in animal husbandry to genetically select genes that are good. Also in breeding of animals, in sex identification. In ecology, it is used in assessment of biodiversity, genetic variations, genetic drift, and also in management of waste, like some organisms, microorganisms can be used to digest waste. In also species identification, that this is a human being, this is an animal, the specific species, like the way humans are Homo sapiens sapiens. And others, we have it being used in phylogeny, where it is the study of the relatedness of organisms, and we build a family tree. Usually build a family tree to show organisms and how related they are. And this is important in animal models, where, especially in healthcare, we want to identify which organism is closest to human beings, so that you can use that particular organism to be able to test maybe vaccines or to test medication before you're able to give them to human beings. These are some of the applications that we have of molecular biology. Thank you for being part of lesson 1 on basics of molecular biology. See you in our next lesson. Remember, subscribe, like, share, comment.